Okay. So first of all, I'd like to thank Gaute and Anton for organizing this wonderful workshop and for inviting me to speak. And in fact, we have several different projects that I think are quite relevant for the topic of the workshop. So I'm really happy that I'll be able to tell you about these results. So I'll just give you a very brief introduction in general about what we are doing and what I'll be talking about. So our general long-term goal is really to understand dynamics and computations in single cells and networks in the hippocampus, and then we are doing that by combining experiments done mainly by my colleagues and modeling and simulations that we do. So today I won't be talking much about this actual like scientific projects, the computations in hippocampus, but instead this will be mostly methods-focused talk. I think I don't have to convince you that to understand the dynamics and computation cells and networks, detailed or data-driven or bottom-up models can be very useful. I mean, that would be like preaching to the choir here, I suppose. So I just say that I think many of us have seen that building high quality data-driven models is a difficult process, and then it is very slow. So for instance, we have been trying to build detailed models of hippocampal cells for maybe 20 years now, and we find that this is a very, very hard process, and you can maybe accept some sort of gradual improvement over time. But one of the reasons that it's so hard is so that initially when we started, we didn't really have the proper tools to develop these and test these models. So we built on earlier and sort of parallel efforts, and we developed optimization algorithms and software tools for model validation, specifically for our purposes. So we hope that by using these tools, we can facilitate in our lab and also around the world a more systematic and reproducible approach to modeling in general. Okay, so I'll start by talking a little bit about parameter optimization. And I'm actually quite fortunate that previous speakers yesterday and today have introduced and talked about extensively about some of the topics that I'll also be covering. So I don't have to spend much time on an introduction. So I'll be quite brief here. So we will be talking mainly about the optimization of detailed biophysical models here. And as you know, many older models were built using ad hoc methods, but more recently, automated methods have become widespread. And just if you think about the method used to optimize neural systems, cells, and networks, several different algorithms have been proposed and then used quite successfully, especially in recent years. But and also in other fields, many other fields outside neuroscience, many other methods for optimization have been proposed. And in general, I think it is really unknown how well all the different methods work on the typical problems that we have in neuroscience. There were some previous efforts that sort of trying to compare algorithms, but it hasn't been done recently, and in the previous work didn't include many algorithms that are becoming popular now. So we did two things here. One is we developed over the years a software tool, which is essentially a user interface to optimization algorithms that allows you to optimize neuronal models or other kinds of neural models as well. And we have developed a set of benchmarks to compare different algorithms on neural optimization problems. So first of all, this is this tool that we now call Neuroptimus. We previously called it Optimizer, but it's sort of too generic. But the GitHub repository is still for in the old name. So this tool, as you will see, first of all, has a graphical user interface that supports a variety of common sort of optimization scenarios for neuronal problems, but it also has sort of batch processing capabilities. You can use it from the command line using configuration files to sort of support sort of more difficult or more other kinds of scenarios. The tool itself supports neuron internally, so it can handle models that are written in the neuron simulator, but it also supports external black box simulators. I will tell you a bit more about that later. Now, at this point, the Neuroptimus includes about 30 different algorithms from 4 different Python optimization packages. These are called Inspired, Pygmo, BluePyOpt, and SciPy. And many of these algorithms can be executed in parallel. Within the tool, there is support for many sort of common paradigms, such as current and voltage clamp recordings. It can handle multiple traces. It can handle time series and abstract or feature data. About the cost functions, we have some internally implemented cost functions, but it also supports the eFL tool for feature extraction that was developed at the Blue Brain project. And in general, yeah, the tool is quite modular, so it's relatively easy to add the new error functions or optimization algorithms. I will just give you a very quick sort of tour of what it looks like when you run this tool in the graphical mode. So this first part allows you to import the target data, which can be a voltage trace, current trace, or features. And then in the second step, you can load your model. Model is defined in Neuron. They can import the model, the HOC files, and then the mod files, and it actually can read the structure of the model, which also allows you to select which parameters of the model you want to optimize. Of course, this approach of directly selecting parameters in a neuron model doesn't work for many other cases, for larger models where you typically have more abstract parameters that you then have to sort of map onto the intrinsic internal parameters of the model. So for instance, if you have a conductance density, then you probably want to set it everywhere in your model, and that's you don't want to do it separately. So you can define functions that map abstract parameters that you optimize onto sort of internal parameters of your model. Then you can sort of define how you want to drive your model. So you can have current or voltage clamp protocols, and then you can have different types of stimuli. You can for running the model. In the next step, you can define and then select features that you want to optimize. So there are some intrinsic early sort of implemented functions that we have within the tool, but you can also use features from eFL here. And then finally, you can select the optimization algorithms that you want to use, and then set some of its parameters. You can also set the boundaries for the parameters, and then you can run your optimization. And the next step, after the optimization process finished, you get your final result, which directly compares the target and the model, and you get the fitness and the parameters. parameters. And then as a next step, you can also get some statistics about the different error functions. You can plot the convergence of the optimization across the generations, and also do other things. So this is what it looks like when you do it from the GUI. Now, when we were trying to compare the different optimization algorithms, then we selected some use cases that are maybe considered typical for neuroscience, and I will show some of those use cases in the next, starting from the next slide. But these use cases are first set up using the Neuroptimus GUI, and then each of these use cases for every algorithm is run multiple times, maybe 10 times, with different random seeds using the batch mode of the software. And then from the results, we extract the evolution of the error, and we do the statistics across the runs. And then we can compare the algorithms in different ways. For instance, one can look at the final error after the given number of evaluations, or one can look at convergence speed, which for simplicity, we just define as the area under the error curve. I hope this will become even clearer as I show some examples. So the first example that we have in our optimization benchmark suite is the Hodgkin-Huxley model. So in this case, we are just tuning 3 conductance parameters of the classic single compartment Hodgkin-Huxley model, and the target data are generated from the same model, but then we sort of forget what the parameters are, and then we try to recover those parameters. This is probably the simplest kind of optimization problem one could think of. What's interesting, actually, that many of the algorithms found it relatively hard to fit this seemingly simple model, and that sort of goes back to what was discussed yesterday about the error landscape of these models. Of course, it also depends on the features that one chooses for optimization, but independent of the features, it turns out that tuning this relatively simple model is not that easy, and we have looked a little bit at the error landscape, and it's surprisingly complex, actually, for the Hodgkin-Huxley model. And then you can see how the different algorithms converged in panel B, and then you can look at the final scores across the 10 different runs in panel C, and you have that. You can compare that for the different algorithms, and the algorithms that we had included some very simple local algorithms, such as the Praxis algorithm that's also used by Neuron, or the Nader-Mead algorithm, also the random search as a baseline, but it also included evolutionary algorithms and particle swarm and ant colony algorithms, and many others that have been popular in recent days, and that are implemented by these 4 packages that we use. Then we have several other benchmarks. So the second one is a voltage clamp benchmark, when you're trying to fit parameters of a synaptic connection based on a simulated voltage clamp experiment. And then a third benchmark is now fitting to actual experimental data from a pyramidal, from a hippocampal pyramidal cell, membrane and neuron biophysical parameters were fit, well, as you can see, maybe in panel C, that this is maybe the easiest problem that where many of the algorithms performed very well. Then going to more complex models, this is now a simplified conductance-based pyramidal cell model, which with 6 compartments. and it's trying to reproduce simulated data from a more complicated pyramidal cell, and it has 9 parameters. So now it's getting more and more complex, and as you can see, now you see more differences between the different algorithms. Some of the algorithms still do quite well, and so some others are sort of mostly failing on this more complex problem. Then a different model class, integrate and fire cell of the adaptive exponential kind. We are now trying to fit both spike times and then subthreshold potentials, and this model has 10 parameters, so this is also a fairly hard problem, and you can see that as that in panel C that not all the algorithms can really do well on this task. And finally, we have a detailed CA1 pyramidal cell model that we are developing in the lab. This has many compartments and many different channels, and this particular version had 16 parameters that we had to optimize. And in this case, you don't see the target data because the target data were actually features measured in experiments, and feature statistics measured in experiments. So that's what the model has to match in this case. And then if you look at all of the benchmarks, then you can sort of make a ranking based on how well the different algorithms did on the different problems. And as you can see, there are some clear winners here, both in terms of final score and then convergence, which includes some evolutionary algorithms and particle swarm, and also among the multi-objective algorithms that are commonly used, the IBL, whatever algorithm is doing relatively well, especially in terms of convergence. And sort of similar to what, for instance, Sharon just showed about side dash, we are trying to develop a database to store benchmarking results, and which would also enable you to do sort of these kinds of comparisons that I just showed. And this would have a web interface, and you could sort of, again, upload your own results, and then those could be integrated in the database, but this is just under development at this point. So the general conclusion about optimization is that we have this software tool which sort of supports a variety of different use cases. It has the GUI, but it also has capabilities for power users. And from the benchmarking, we find in general that local algorithms are like the Praxis one, which is implemented by Neuron, probably not suitable for more complex tasks, but you do find a whole bunch of algorithms that perform relatively well across the board. And these include some evolutionary algorithms, particle swarm, and also some multi-objective methods. Okay, so let me switch gears and talk a bit about model validation. So here, again, I'm in a relatively easy situation because Sharon beautifully introduced model validation, and we will use the same framework, as you will see in a minute. So the starting point for our work was that, well, a large amount of experimental data are available for many cell types, and that includes the CA1 pyramidal cells, which we are especially interested in. There are currently more than 100 models of CA1 pyramidal cells in ModelDB, but as has been pointed out, most of these models have been custom built for a particular purpose. They use a very limited part of the available constraints. There are a large amount of experimental data, but each of these models uses just a very small part of this data. It's mostly unknown how any of these models would behave outside their original context. This also could be the reason why model reuse is relatively limited. And I should note that even when model reuse has been attempted, the models are often sort of retuned to match some new piece of data. And this often leads to what's called regression in software development, where when sort of trying to match new targets actually leads to the model sort of breaking down on things that it was good at before. Now, since testing the models and comparing models manually is very difficult and also error prone, we agreed that we needed automated validation suite. Fortunately, we already had a good starting point with HippoUnit, and we also took inspiration from NeuronUnit. So our framework is based on HippoUnit. It uses the same sort of framework and same ideas, which I really don't have time to talk much about, but Sharon actually talked a bit about this as well. So our test suite is called HippoUnit, and it aims to validate detailed hippocampal CA1 pyramidal cell models. It has simulations that is based on simulations that mimic reported experimental protocols in different types, and I will talk a little bit more about that later. The tests are implemented in Python, and one preferred way to run them is from Jupyter notebooks. Once the simulations are run, features are extracted using eval or functions implemented by HippoUnit, and then the observations, the experimental data are compared to the model predictions, and we often use feature-based error functions, which just means that we look at the features, look at the mean of the features in the experimental data, and we measure the model performance as a distance from the mean in units of the standard deviation in the experimental data. And then the suite, the HippoUnit produces output, which includes the individual features, the feature scores, and then the final score, which is the sum of all the things that are useful to understand what's really going on, which includes figures and feature values in different forms that you can then use to understand what's going on. One thing that I should note is that HippoUnit can be used for anatomical and biophysically detailed models, and then at this point, the model has been used to test models. The software has been used to test models implemented in Neuron. For that, one needs sort of standalone or pure versions of the models, which have the necessary HOC files and mod files, and including the morphology and the biophysics, but it shouldn't have any GUI or built-in simulations, runtime modifications, which are actually quite common if you just download download a model from ModelDB. So we had to prepare these pure models and then check that the behavior is identical to the version which is in ModelDB. So then we have, and this is the work of my graduate student, Shara Shari. She implemented, at this point, several tests. I will show 5 of them here, just very briefly. And some of these tests are about the response of these neurons to somatic current injections. Some others are about in the propagation and integration of signals within the dendrite. As a start, we took a bunch of models from the literature, which have been quite widely used and are well known, and we prepared the pure versions, and we used them to sort of test this HippoUnit validations. So for instance, this is the Simatic Features test, which uses the response to somatic current injections, and from the current injections, from the responses of the models, the current injections, we extract features, and we extract the same features from the experiment. And you see some of the examples of the features that you get, which include things like action potential height, action potential width, spike count, and many other parameters, only just a few of which are actually shown here. And you can compare the behavior of each of the models to the experiments, which is shown by the crosses in each case, and the different models are shown by the different colored dots. And then once you have the raw features, you can calculate the feature scores, and here you have some of the feature scores aggregated across the different current injections. And one important point is that sometimes it's the models cannot be evaluated on all of the features, and that's also something that we take into account. Okay, I think I need to speed up a little bit. So probably won't have time to explain in detail all of the tests, but this one, for instance, looks at the amplitude of backpropagating action potentials into the dendrites. The next one looks at the attenuation of postsynaptic potentials when there is a synaptic input to the dendrite, and one can measure the attenuation from as the signal reaches the soma and compare it to experimental data. Then this one looks at a depolarization block, which is about looking at what happens as you increase the amount of input, amount of current that a neuron gets. And for most neurons, eventually the neuron will stop spiking as it receives more input and goes into depolarization block, which is sort of a state where the neuron is not excitable anymore. And one can again compare the behavior of the models with the measured data on depolarization block in CA1 pyramidal neurons. And as you can see, most of the models actually cannot reproduce depolarization block. And actually, some of those that seem to do it are actually, it turns out they are actually cheating the test. So it also happens. Tests are not perfect. So sometimes we find examples when a certain model can sort of cheat the test and sort of convince the tool that this is actually producing, in this case, depolarization block, whereas in reality it's not really doing that. And this last one I wanted to show is maybe the most complex one, which is looking at the nonlinearity of synaptic integration in the dendrites of these neurons. And this is a case which is where you go through different dendrites of the same model cell and look at whether the response to a different tests, and you can get sort of a characterization of the different models. And on the right, in fact, we have our model that it's under development in the model, which we are using these optimization tools and validation tools that I just mentioned. So it's not perfect yet, but I think we are getting better. Well, I gave you just a few sort of final thoughts. One is about HippoUnit, as you have seen, can be used to evaluate and compare existing models, which is great because you get an independent and standardized verification of the behavior. One can learn more about models published by other groups and maybe decide which other models could be used for your purposes. And during model development, you can easily evaluate after every iteration, and you hopefully can avoid regressions. And it may also enable the creation of sort of community models where you can sort of iteratively refine models, maybe in collaboration of different research teams. The HippoUnit the tests have been integrated into the eBrains validation framework. They can be applied to models in the model catalog, and the results are searchable. There are maybe a few important, these are, I think, are important things that I need to point out, that if a particular model gets a high error score on a test, that doesn't necessarily imply that the model is bad. It could be that the data are to blame, really, because they're noisy or biased, or maybe the test has a bug and it doesn't work properly, or the model was simply designed to mimic some other experimental conditions, another species, strain, other temperature, other recording technique, and so on. And of course, even larger error scores, should be considered normal, because the real neurons are also diverse. They are not like an average neuron. So even large scores could match some members of that population. And finally, the model could also be, of course, if it fails one test, it could still be great for other purposes. It's important to say that validation should not be used blindly. So one should consider the modeling goals, which many models, some well-designed standardized model formats could be really helpful. Just some final comments. One, about multi-purpose models. As you can see, there are tools like ours, like these validation tools, allow you to test the behavior of models in many different situations. Of course, what is a good multi-purpose model? A good multi-purpose model is one that makes good predictions in many different situations. And of course, this is also analogous to sort of good generalization in many cases. So, and it could be different forms of generalization, from performance and just untrained features all the way to performance in new paradigms. I think there are a bunch of important lessons one can learn from machine. You want to validate and ideally also train in all the domains where you want to make predictions, because interpolation is in general much easier than extrapolation. And you don't want to use all the data for training, because you also need the data for validation to avoid overfitting. This also allows you to sort of select models that make good predictions. I think I will just skip this, because maybe we can come back to the issue of collaborative modeling and community models during the discussion. And I will just thank my current and then previous students in the lab, and also the collaborators. And I thank you for your attention.